Welcome, everyone. Hopefully, we'll have still a few more coming in, but I will introduce our speaker for the day. Very, very lucky to have Dr. Eva Stukenbrock joining us. And their research focuses on the ecological interactions and coevolution of fungi associated with plants. Since her PhD at the ETH in Zurich, she has used the plant pathogenic fungus Zymoseptoria tritici as a model to study pathogen evolution. During her postdoc at Aarhus University in Denmark, she worked with Professor Michael Schurup to apply whole genome coalescence analyses to infer the speciation history of Zymoseptoria tritici and related species. Then in 2010, she was appointed a group leader at the Max Planck Institute for Terrestrial Microbiology in Marburg, Germany. And since 2014, she is a Max Planck Fellow and professor at Kiel University. Her group integrates computational biology with experimental and molecular approaches to study mechanisms of host specialization of plant pathogens. And I will just say she has got some fabulous papers published. So really excited to have this guest. So I'll turn it over to you, Eva.
Yeah, thank you very much for the nice presentation. I will just and for inviting me. So you see my screen?
Yes.
And it's in presenter mode. Excellent.
Yes.
Good. Yes, thank you for the invitation. I'm very excited to join you online. Would rather be in Canada, but it's good to meet you online. I'm going to start by just making this a bit more simple title. I sent Maria-Lena a terribly long title here. But essentially what I wanted to express here is that, as Matthew mentioned, for many years we've been interested in the evolution of fungal plant pathogens, and we've been using genomics, transcriptomics. And in the last couple of years, and we've been trying to use some of these new deep learning approaches that are available that allows us to predict structures of proteins, to ask some of our old questions about species evolution with these new tools. And Matthew also mentioned already Zymoseptoria tritici, which is a very important pathogen of wheat, in particular here in Northern Europe. And it's characterized by producing these necrotic lesions and small black dots here that are the asexual fruiting bodies of the pathogen. So one of the questions that I've been working with since I was a PhD student many years ago, and just for you to think about that, actually now many of you are doing your PhD now, that you can actually be stuck on the same topic for many years. But I've been interested in the evolutionary history of Zymoseptoria tritici. And back then, during my PhD, we used a population genetic approach with collections from all over the world to identify the center of origin of Zymoseptoria tritici. And this turned out to be the Middle East. We also used different types of evolutionary analysis, coalescence analysis, to determine when Zymoseptoria tritici originated as a separate species. And this gave us some hints that this pathogen has evolved with the domestication of wheat and subsequently also has been spread with wheat cultivation worldwide. So we have been working with excellent phytopathologists in Iran ever since, who have been enabling us to work with collection of Zymoseptoria from the center of origin of Zymoseptoria tritici, but also the center of diversity of this group of pathogens. And so what's wonderful about working with colleagues in Iran is that they provide us with access to these wild relatives of wheat and barley, which have symptoms of Zymoseptoria pathogens. And so we've been using such collections here to further investigate the evolutionary history of these pathogens here. And so over the years here, we've been describing new species of Zymoseptoria on different grass hosts. And we've particularly been focusing on Zymoseptoria tritici, as I mentioned to you before, and lineages of Zymoseptoria tritici that infect wild wheat relatives, species of Aegilops. And then we have another pathogen which is much less known, Cymoseptoria passerini, that is a pathogen of barley, a very rare pathogen of barley that also is found in barley fields, for example, in North America, but not as prevalent as Cymoseptoria tritici at all. But we find also here in wild barley, we find relatives of Cymoseptoria passerini. And so these two species here provides us with a very powerful system to address more recent evolutionary changes and evolutionary changes that have occurred over longer timescales. So one of the great things about the system here is that we can take these isolates into the lab and then we can inoculate them on different hosts to assess to which extent this host specificity also is that we see at the sequence level also is reflected at the ability to infect different hosts. And I'm showing you here for Cymoseptoria tritici and Cymoseptoria passerini isolates that we have from the wild and domesticated host, and how we can really demonstrate here that the recent divergence of these pathogens here on closely related hosts indeed reflect a degree of host specificity. And so for us, an important question is, so what is it that confers this host specificity? And Wagner Fagundes was a PhD student in the team. He's a postdoc now working at the Max Planck Institute in Tübingen. But he took out a population genomic data set of Cymoseptoria tritici from wheat and from these Aegilopsis wild wheat. And then he in particularly focused here on the populations from Iran, which essentially are geographically coexisting, but are infecting different hosts. This is a split, the diversity of these isolates here illustrated as with a split tree analysis. But we can also look at the divergence of these isolates here in a PCA analysis. And so what you will see is here that we have the isolates in Iran from wheat clustering very closely together. And then from Aegilops, we have these two distinct populations here, which was something that we first were wondering about what this actually reflect. And then we went back to the original material from which these isolates were derived, and could actually identify that these two Cymoseptoria tritici populations, they reflect different host-specific populations from two Aegilops species, Aegilops cylindrica and Aegilops tauchi. So again, here really reflecting how the adaptation, the specialization to different hosts is an important driver of evolution of these pathogens here. And so many of you, I know you're also interested in fungal evolution and adaptation. And I think really one of the big challenges we have when we work with this type of omics data, population genomic data, comparative genomic data, is to distinguish neutral variation from the variation that is selected. And again, here our question is really what has been selected during the specialization to different hosts. And this is a plot here along chromosome 2 here of variation in Cymoseptoria tritici. And some of you who work with this type of data will also recognize there's a lot of variation, and the level of variation varies along the chromosome. And how can we identify what is really here under selection? I don't want to go into details here, but what we have been doing is we've been taking different approaches to identify signatures of more recent selection, so-called selective sweeps. So here you can use different statistical approaches to identify where do you really have outliers in the genome, where you have specific patterns of genetic variation that indicate that here you've had a strong impact of positive selection. And what I'm showing you here is a plot along the 13 core chromosomes in the two lineages of Cymoseptoria tritici from wheat and from Aegilops. And essentially what you see here are sort of outlier regions here that with two different methods for identifying these selective sweep regions here come out as being statistically significant. And so what Wagner he had here was a small set of genes where we have evidence for them being under strong selection and being our candidates for explaining host adaptation. And I want to focus here on one region that we were particularly interested in here on chromosome 7. So this comes out in both the Aegilops and the or the wheat population as being a region here that has been under strong recent selection. And if we zoom in on this region here on chromosome 7, then we can look at different other measures of sequence or of parameters here. The DXY as a measure also of genetic variation, FST, the measure of genetic differentiation between the populations, and then the nucleotide diversity across the sequence here. And interestingly, also in this region here on chromosome 7, the selective sweep region here, there are 3 genes that encodes small secreted proteins. And I'm sure that many of you, like me, we love small secreted proteins because these are our putative virulence factors. And so we've been very excited here about identifying such putative host specialization genes in this selective sweep region. So what's the next thing to do here? That's, of course, to further validate the relevance of these by doing a knockout. And with Cymoseptoria tridici, so we focused on one gene here, this 00299. And one reason that we focused on 00299 was that this gene has actually popped out in another analysis that we had been doing, and where we had been comparing gene expression across Cymoseptoria tridici and Cymoseptoria pseudotridici and Cymoseptoria adabilia during host infection. And one of the genes that came out here in this experiment here by Janine Houweisen, so very briefly what she did here, she would infect these different isolates of Cymoseptoria tridici and of pseudotridici and adabilia on wheat, and then she would assess gene expression at different time points. And the number one effector candidate that came out as being most strongly upregulated during early host colonization was this 00299. So we were pretty sure, okay, so here we must have a very strong determinant of host specificity. So we can do gene deletions, we can do genetics in Cymoseptoria tridici, and Janine, she did that. And this is the result. So the phenotyping in planta. So we have 2 measures of disease, and one is the proportion of the leaf area that is necrotic, and then the density of these small black dots that are the asexual fruiting bodies. And what you can see here is actually a little bit of a boring result, because essentially the mutants of this gene here appear to be either as virulent as the wild type, or to some extent maybe even a little bit hypervirulent, but there's some variation between 3 independent mutants, and so it's not really something that we are very exciting about. So in Janine's transcriptome analysis, there were a number of other genes that came out as being very highly expressed. Some of them also in Wagner's analysis showing signatures of being under selection. And we deleted these genes here. And I just want to mention that for Cymoseptoria tridici, we don't have a CRISPR-Cas protocol working yet. So we've been using Agrobacterium tumefaciens mediated transformation. And I want to show you here the result. I will go a little bit fast over it because it's very boring results. But this essentially show here for 5 other genes that we deleted. And I could show you more of these highly expressed small secreted proteins that are diverged between species. They have actually all the signatures of being effector candidates, but when we delete them in Cymoseptoria tridici, we don't see any strong effect on virulence. And sometimes, so you can even here, Janine, she tested here 6 independent mutants, nothing came out. So she did really a very proper work here. And so this has made us also speculate about the way that we are doing the phenotyping of these effectors. What is actually defining host specificity? Are we actually looking at the wrong phenotype here? That's one thing. And another thing that, of course, also comes to one's mind is that there must be also a redundancy in terms of effectors, so that there's not one protein that defines host specificity, but several. And in addition to that, this thing of if there is a redundancy of effectors, then perhaps the best approach is not to use gene deletion to study if functional relevance of these effectors. So at the same time as we moved on and sort of are thinking about the relevance of these effectors, there was some work that came out from the group of Bart Thomas some years ago. And Bart had also been working for many years on an effector candidate where he just could not figure out what is the functional relevance of this effector. And one thing is that he had a big problem of expressing this effector in bacteria. And this led actually to the discovery of this effector being an antimicrobial effector. So it doesn't play a role in interfering with immunity, but it plays a role in interfering with the microbiome of the host. And so that also made us think about that we, for Cymoseptoria tritici, for several other plant pathogens, we only know the immune interfering function for a relatively small number of these small secreted proteins or effector candidates, and that there may be a very important relevance of these proteins here in terms of interacting with the microbiota, and thus still a lot to be discovered. And so the question that we move forward with is if some of these secreted proteins, a certain proportion of them, actually are involved in pathogen microbiome interaction, and could there potentially also be a relevance in terms of the host specialization for such antimicrobial or such microbiome interactions? So the new questions that we have been moving forward with here is that effectors play a role not only interfering with immunity in the host, but also to antagonize microorganisms. That's something that we had been wondering about for many years, is why these immune interfering proteins, when we look across very diverse pathogens, they are so diverse. How have these functions evolved? And of course, the hypothesis is that they may have actually evolved from antimicrobial functions or antimicrobial effectors. And with respect to host specialization, we have been thinking if these effectors also may evolve in response to a certain host microbiome. And this last sort of hypothesis or question here is actually the one that I would like to just show you how I've been actually thinking about this, or how we've been addressing that, because an important assumption is, of course, here that different grasses that are host to Cymoseptoria would host different microbial communities. So some years ago, we actually set out to address this. So we took 3 different species of wheat and we propagated them in the same soil. And then we assessed the diversity of bacteria and fungi. And first of all, one thing is that the strongest explanatory factor for microbial diversity in a plant is actually the tissue. So you find a leaf-specific microbial community, a root-specific community, and a rhizosphere-specific community. So just by looking on all the data that we generated, we didn't see a clear signal here with respect to host specificity. But if we zoom in on the different compartments here, then we can actually see a very clearly distinct communities depending on the triticum species from which these communities are derived. And I would like you then to think about that a Cymoseptoria pathogen that goes into the leaf of Triticum aestivum will encounter a different microbial community if it would be infecting Triticum boeticum, that is a wild wheat relative. And I'm showing you the data only for the bacteria here, but with the ITS sequencing, we find the same pattern for the fungal community. So the microbiome of these wheat species also are distinct. So again, thinking about host specificity, At least is a difference here that these fungi have to cope with when they infect their host. So we have been interested in addressing this specialization to different microbial communities. And we have been, as I've been mentioning several times, we've been working in this genus Cymoseptoria. But of course, one consideration here was if we can really identify a signal of specialization to the microbial communities within a genus. And therefore, we are also looking at the microbial communities of Beta vulgaris and its interaction with another dotidiomycete fungus called Cercospora peticula. And then we've moved outside of the dotidiomycetes to look at the Pyronophora teres, that is also a host of barley, with the aim of looking at if Cymoseptoria passerini and Pyronophora teres essentially are specializing to the same microorganisms in barley, or if there are different patterns of interaction with barley pathogens. So this is a sort of a framework that we have been embarking on here. And I will come back to Pyronophora teres and Cercospora peticula later, but we will still stay a bit with the Cymoseptorias for now. So we've been taking this comparative approach here, and we've been focusing on the proteins that are secreted. And this is work done by Thais Del Sasso, who is originally from Brazil and now a postdoc in the team. And Thais is really the person here who has been diving into these new deep learning approaches here. And this I'm sure that maybe some of you have even been playing around with AlphaFold or other tools here for predicting the structure of your favorite protein here. And our goal has really been to do this at a genome level and also at doing it at a comparative genomics level, if you can say so. So as an evolutionary biologist, why is it? What can we use these protein structures for? And first of all, we can learn something about the evolution of proteins, because we can, as I mentioned to you at the beginning of the talk here, we can actually do structural alignments and thereby learn something about how proteins have evolved really at the, let's say, at the 3D level, where that define also the functional properties of the proteins. In addition to that, for many of these effectors, these small secreted proteins, for many of them, we've been walking in the dark because we've had no clue about what these proteins are doing, because many of them do not have any conserved protein domains. There's no similarity to anything in databases. However, based on having these structures, this allows us actually to may allow us to identify conserved protein folds, and thereby to have new insights into putative functions for some of these proteins here. So what Thais, she has done is that she has set up a pipeline here where she has been focusing for the different fungal species here on their secretomes. And then she's been using some tools here that one of them, I believe some of you will know, effector-P, which is the previous version has been based on sequence. It's also a deep learning tool, but it's based on the protein sequence and recognizing different features that may render or may, let's say, provide a probability or allow us to recognize a probability that a certain protein may be interfering with the immune system of plants. And now there is a new version of effector-P coming out, which is also incorporate information about the structure of the protein. So that's, I think it's going to be even refined more what we can do with this. And then there is another tool here that come from Bart Thomas' group. Fantin Mesny is the postdoc who has developed this. And it's a tool that's based on protein structures. and predict if a certain protein, based on its structure, has antimicrobial properties. So we have now the possibility of identifying or predicting if a protein may be immune interfering or antimicrobial. And in addition to that, we have, of course, expression data that also is very important for us to consider the functional relevance. And as I mentioned to you, we may have new insights into putative function based on conserved protein folds here. And last but not least, as I mentioned to you, what Thais is also working on now is really to align protein structures to learn something about how certain protein folds may have changed, providing new functions to the protein. So one thing, if some of you are playing around with these tools here, I think an important figure, which it's a little bit boring because it's technical, but it's showing the probability whereby a confidence score, how well we can actually predict the structure of a protein of different length. And what you can see here, and this is still actually data from AlphaFold 2, so now we have AlphaFold 3. But the pattern is the same, is that where we have the lowest confidence score in our protein structures are really for the small proteins. So there, I'm sure this is something that's going to develop very fast, that we will have more, we will have also more or better data to predict better the structures of the small proteins. But this is something to keep in mind, because many of the proteins we're interested in are indeed these smaller proteins here. So we have to think about also confidence score of the data we're working with. But the wonderful thing is that if we take the secretome of Cymoseptoria passerini, we can actually predict the structure for 90%. And that's with AlphaFold 2, and I don't have a figure for it, but I can say with AlphaFold 3, this increased to 95%. So we really have now some new insights into this secretome. So I'm going to, on the next slide, show you a little bit of a busy slide. And so I just want to explain to you how to read it. So this is, we have the protein structure, and then we can compare structures, or we can identify structures that are similar, that share the same protein folds. So you could, with the terminology from sequence alignments, you can say these are proteins that we can align, or we can generate an alignment and make comparisons. But so essentially, this is a network or a cluster of 6 proteins that share the same structure. And here the width of the bands that connect them indicate how similar they are. And then you will see they have different colors, and that relates to some of the annotations that you saw before, that we have this possibility with effector P to predict if a protein is an apoplastic effector or if it's antimicrobial or non-antimicrobial. So the antimicrobial proteins, the predicted antimicrobial proteins have this blue circle here. So if we do this exercise for the whole secretome of Cymoseptoria passerini, we can identify 72 of these clusters of protein that share a similar protein structure. And there's, of course, some proteins missing here, and those are the singletons. Those are proteins where there's nothing similar in the genome of Cymoseptoria passerini. The colors here, as I mentioned to you before, indicate the predicted functions. And one thing that you immediately should see here is that there's a lot of blue colors. And what we find here is actually that almost half of the secreted proteins are predicted to be antimicrobial. And so I'd like to just to step back and think about that. So that means that if this is true, if this prediction is true, it means that half of the proteins that the fungus secretes somehow engage in interaction with other microorganisms. And so I think that's a lot. But that, of course, and that's a lot. And it also means that that's an important part of the biology of the fungus. Again, it's a prediction, and of course, something that has to be validated. But with this type of, let's say, clustering and overview, we can identify protein clusters that are enriched in these antimicrobial proteins. For example, this cluster here of chloroperoxidases. And before we didn't, we did not recognize these as being chloroperoxidases. And these chloroperoxidases are known to be to have antimicrobial functions. So indeed, here the prediction at least is sort of indicating that there is something to it. In the same way, we can also identify clusters that are enriched in putative real immune interfering effectors. And actually, this cluster here includes also this PUD1. It has a name because these are homologs, structural homologs of effectors described in other plant pathogenic fungi, in Verticillium and Alternaria plant pathogens. And in this cluster, we also find a homolog of this 00299 effector that I showed you before. So I would like to focus together with you on this cluster 12 here. I just want to show you one thing before we move on with the structures, and that is actually the alignment based on the protein sequence. And this is a mess, right? And before we had never considered that these 9 proteins would in any way be related to each other. But now where we have the structures, we can align them. The protein sequences only include 2 conserved amino acids, 1 cysteine and one tryptophan. So just to indicate here the power of doing these comparative analysis based on clusters. Okay, so here looking at this G12 cluster here, you can see it has a lot of purple and orange here. And this G12 cluster here is together with 5 other clusters. We find them to be enriched in predicted effector proteins. And so this also allows us now to zoom in and refine our hypothesis related to what are the proteins that are involved in interfering with the immune system of the plant. And we even have a predicted function here, this protein is categorized as a so-called killer toxin family 4 protein. Killer toxins are known first are described in yeast and in other fungi, and are also known to have antimicrobial properties. There's another cluster here that we'll come back to here, and that is also a family of killer toxins. This is killer toxin family 6, so the KP6 effector proteins here. And these are proteins that we also, based on gene expression patterns and for other reasons, we're interested in, and that we picked up for more functional validations. And so in terms of functional validation or of functional properties of these proteins here, then at first one can ask the question also, so we're still just addressing here Cymoseptoria passerini. So why would the pathogen have these 9 highly similar proteins? And the different colors indicate that these different proteins, albeit their similar structure, have different functions. And when we overlay here the structures here with gene information about the expression of these genes, and the way that you should read this, so here we take gene expression data from our RNA-seq data, and now the colors inside the circle is no longer related to the annotation, but the extent of expression of the genes at 4 days post-infection, 7 or 10 days post-infection. And what you can see is that different proteins are expressed at different times of the infection, suggesting that although they have the same structure, there is some differentiation that may relate to functions at different times of the infection. Okay, so we wanted to use the killer proteins to further validate some of these predicted functions. And here I'm going to make a jump from, now you've been looking at data from Cymoseptoria passerini, so much of the experimental work we are doing or have been doing is based on Cymoseptoria tritici. That's really been our workhorse. So I would like to show you some of the data we have from these killer proteins from Cymoseptoria tritici. So don't get confused here. So we're working across both systems. And I already showed to you that how can we now validate functions? And one thing is you can knock out the genes and do mutant phenotyping. And this takes a lot of time, and so far also has not been really let's say, the convincing method. And so instead, what we've been focusing on here is to take our Cymoseptoria proteins and then express them in a heterologous system in order to address what these proteins are doing. And one approach in order to see if they may have an effect on the immune system of the plant is to express them in a plant. And then we also have been expressing these Cymoseptoria proteins in other microorganisms, in bacteria or in yeast, in order to address if they may have these antimicrobial properties. And so I would like to focus here on these two approaches here. And so now I don't know how plants, how many of you are plant experts. This figure here is from now it's actually an old review, but it's a simplification of the immune system of a plant. And very briefly here, to those of you who are not working on plant pathogens, but a plant has receptors that are acting outside of the cell or inside of the plant cell to sense when there are foreign molecules. And these foreign molecules can, for example, be elicitors produced by pathogens, so-called, or it can be flagellin or chitin, so-called pumps that can induce an immune response when they are recognized in this intercellular space. And what happens when this occurs is that the first immune response of the plant cell is to produce reactive oxygen species. And so what the fungus does is that it secretes effectors, and these effectors can act either outside of the plant cell or inside of the plant cell. And you will remember before on the previous slide, you would see these cytoplasmic and apoplastic effectors, depending on where the effectors are predicted to act. And essentially the role of these effectors is then to silence this immune response. So we can actually measure, what we can do for example, let's say in our toolbox or in our model, experimental model, Nicotiana benthamiana, which like Arabidopsis thaliana can be a model for molecular analysis in plants, is that we can activate an immune response by treating the plant with, for example, chitin or with flagellin, and that will elicit such a production of reactive oxygen species that we can measure with a so-called luminol assay. But then what we can do in order to assess the function of our effectors is that we can express the effectors in this plant tissue, and then we can actually observe if we have a reduction of the reactive oxygen species, indicating that there is a suppression of this immune response. So this may all be familiar to some of the plant persons in the audience, but to others, this is the method that we use here to assess if effectors from Cymoseptoria may interfere with this basal immune response. And so we took, in the first place, we started with 4 proteins that are highly upregulated during early host colonization of Cymoseptoria tritici, 1 KP4 protein and 3 KP6 proteins, and then we express them in Nicotiana benthamiana to see if they interfere with this ROS production. And so the way that you should read this is, so here we have sort of our control where we don't expect any interference in this ROS burst that we see. Then we have 2 effectors that we know can suppress this reactive oxygen production. And you will see here that relative to this sort of standard here, we have a reduction of reactive oxygen species production. And then we have 2 other effectors here that do not interfere with this ROS production in Nicotiana benthamiana. And when we take our three killer proteins here, then we can actually see that we have one of the KP4 and one of the KP6 effectors, where we also see a significant reduction here of the ROS production, and maybe also one for a bit for one of the other KP6 effectors here. But we do see that we can use this assay here to identify immune interfering effectors. So this is our assay here for assessing immune interfering properties. And then, as I mentioned to you, that we express these effectors also both in bacteria and in yeast. And one way that one can do this, and that we started out was expressing these effectors in E. coli under an inducible promoter, and then see if E. coli can actually grow in the presence of these effectors. And our hypothesis here is that if we see a reduction of growth when these effectors are produced, then it could be indicating that the effector is antimicrobial. And so what we interestingly see here is that for two of these KP6 effectors, we do see a reduction of bacterial growth, which may indicate that these effectors are antimicrobial. And so the interesting thing that I have highlighted down here is that these two killer protein effectors that do not severely interfere with bacterial growth, they are actually the one that most strongly inhibit the immune response in Nicotiana benthamiana. So just by looking at these four proteins, we do see some differentiation in functional differentiation here. And so what the killer proteins here already give us some indication is that based on conserved protein structures, proteins belonging to the same family, they may evolve different properties relating to interfering with bacteria in the plants or interfering with immune responses. And so, of course, we would like now to understand what are exactly the properties here that confer these differences. So one thing also E. coli is a great bacterial model system to work with, but there is not E. coli in the plants. And furthermore, there may be other, when you should express a fungal protein in E. coli, there may be a lot of, let's say, other problems in terms of the processing of this eukaryotic protein in the bacterial cell. And so therefore, we have moved from expressing proteins in E. coli to expressing proteins in a yeast, Pichia pastoris. And the great thing is that we transform the yeast cell. And then what we can do is that we can plate out our bacteria of interest, and then we can add our Pichia, our yeast cells here, expressing here either GFP as a negative control or a Cymoseptoria tritici effector of interest. And then we can look for an impact of the effector on the growth of the bacteria. And as you see here for this example here, then there is a halo of the bacteria indicating that this is an antimicrobial effector. So this is a screen that we have tried to establish here for expressing multiple Cymoseptoria tritici effectors as a screen for antimicrobial properties. And the beauty of having these effectors expressed in Pichia is that one can do the next steps and then actually purify the proteins for more clean assays of antimicrobial properties. And so what we in addition to that, have been doing, in addition to establishing our Pichia expression system, is that we have generated culture collection of bacteria as well as fungi from wheat and from barley. And this is just to give you an overview of the diversity that we have. We have actually several thousand and several hundred bacteria and several hundred fungi in culture that we can then use for our assay with Pichia. So Leonard, so what I showed you before was really was four proteins, three KP6 proteins and one KP4 protein. And Leonard Paschinger, who is a PhD student in the team, so he went actually and screened all of the killer protein 6 and killer protein 4 proteins in Cymoseptoria tridici with these two assays, this expression in Nicotiana benthamiana and the Pichia expression here. And what you can see here is that we find we find one other KP6 effector that interfere with immune signaling in Nicotiana benthamiana. And then we do find that several of these KP6 proteins here, they interfere with the growth of bacteria from the microbiome of wheat. And in the same way and so just to mention here that we have been playing around here both with purified proteins, but even if we just take the supernatant of the yeast colonies, then we can also, in the plate reader, we can also demonstrate this antimicrobial effect by growing the bacteria with the purified protein or with the supernatant from the yeast expressing the effector to assess here the impact on the growth of certain specific bacteria. So and as mentioned, so this is for the KP6 effectors, and in the same way for the KP4 effectors, what Leonard can show is really this amazing differentiation in the properties of the function of these structurally similar proteins with respect to immune interference and antimicrobial properties. And so the take-home message from this is really that we first of all, we can validate here for our KP6 and KP4 effectors. We can validate this prediction of antimicrobial properties as well as also the immune interfering properties. And it also suggests that there is a very high extent of functional divergence around conserved protein folds in these Cymoseptoria pathogens here. So one of the questions that Thais, she was interested in understanding as well. So if we have a conserved protein structure and you have two similar proteins, one is antimicrobial and the other is interfering with immune signaling. So are there some properties that we can recognize that render that is sort of more associated with one or the other function? And so what Thais did was that she took these proteins from the secretome of Cymoseptoria pessarini, and then she categorized them according to being purely effector proteins, non-antimicrobial, or having this dual prediction of being antimicrobial and immune interfering, or being only antimicrobial, and then anything else that is non-antimicrobial and non-immune interfering. And then she has been diving into some of these physicochemical properties of proteins. And so here we are really moving on, let's say, on very new land for us, because we're not biochemists in the team, but we have learned a lot here by comparing different parameters. And I just want to show you here for one of the physicochemical properties that we can really where we can see some difference, and that relates to the hydrophobicity of the proteins. And so we know that hydrophobicity is very important in terms of the affinity of a protein with, for example, membranes. So how easily it interacts with membranes. And what we find here is that for these four classes of proteins, then we can see that the effectors, so the immune interfering proteins, including also those that may have an antimicrobial function, they have a different pattern of hydrophobicity compared to the purely antimicrobial or other secreted proteins. So we do think that some of the functional diversification that we see, some of the evolutionary changes of these protein folds relate to physicochemical properties as hydrophobicity. And so just to sort of, if this is a crazy box plot here. These are all of the 72 clusters that you've seen on the previous slide here. And comparing here the hydrophobicity, and you will see here these killer protein that have a relatively low extent of hydrophobicity compared to, for example, a cluster of proteins which are encoding purely antimicrobial proteins here. So this is something that we are exploring further and also trying to understand better how we can really disentangle what are the changes that confers these different functions of the proteins. Okay, so to summarize the first long part here, we do find that many of the secreted proteins are predicted to be antimicrobial, and we do actually validate this for several of them. We believe that the differentiation of proteins around common folds do reflect this adaptation to the host microbiota as well as plant immune receptors. Yes. Okay, so the last minutes here, the last 5-10 minutes here, I would like to go back to the question. So now we've been looking within the genome of, is it already 50 minutes now? Because I can stop now.
It is
I
50
think
minutes. We're
I
flexible.
think it's 52 minutes. Yeah, good. So I think I will stop here. So I had a small. I'll just show one slide here. I just want to take you to this slide here, where I want to say that what we are doing now is that we are moving from looking at the 3D secretome of one species to the 3D secretome of 9 different genomes, and trying to make sense of that. And so this is then a story I will keep for another time. But yeah, so thank you very much, and I'll just go quickly here to the acknowledgement slide here. So thank you very much.
Thank you, Eva. That was wonderful. That does sound like a story that would be hard to fit into a few minutes.
Into 5 minutes, yeah.
Okay, that was fabulous. Well, I think you've gotten the background that normally we have a couple of questions from the audience, and then everybody who's not a fellow leaves, and the fellows remain for some broad and extended discussion. So maybe we just first open it up to non-fellows who might be in the audience to have a few questions before we leave the fellows for their conversation. While people are formulating, maybe I'll just start off with one question, which is I wonder whether the prediction of antimicrobial action might also indicate some targeting of plastids within the plant cells, since they're microbial in evolutionary origin and have still some prokaryotic-like features. Could there be some, misclassification for effectors that might target the plastids? What do you think about that?
I think that's a very good point. I had this question a bit in a different way before, where somebody was asking, well, if you have a predicted cytoplasmic effector that is also antimicrobial, how can one explain that? That you have something that's antimicrobial inside the plant cell. And so I think that's a good point, that there might be some that indeed that some of these properties that we do predict here actually may relate to interference with plastids. So that's a good hypothesis.
Can I ask a question?
Yeah, I'm not sure what kind of view I'm in. I can't see anybody else. So if you want to talk, you just have to talk, I think.
Great talk. I mean, using structural-based analysis to understand function is a fantastic way. We're doing quite a bit of that stuff. I'm just wondering, with respect to putting the structure into clusters, have you taken into account, because we noticed that if a protein has a lot of intrinsic disordered regions, it is very promiscuous. It interacts with many, many proteins. So how do you, take that into account when he's putting the clusters of protein together as a group?
So how we account for
Yeah, we
these
noticed that it's
disordered
actually
regions?
skews up the analysis quite a bit.
Yeah. Um, so you mean how we can group them if they have like these disordered regions or
Yeah.
yeah. I mean, so there is of course a threshold. So you set some threshold for when you group something together into a cluster. And indeed, this is something that we've been thinking a lot about.
Mhm.
These so indeed that depends on where you set the threshold. And indeed, these disordered regions that often are very difficult also to predict. Actually,
Yeah.
the example that I was going to show here the last 5 minutes. So these are two proteins from one from Cercospora and one from Cymoseptoria that are almost identical, when you overlay them, but then they just have one has this strange disordered region, and they
Mm.
show very different properties in terms of inhibiting certain bacteria. So I do think that this is something that's where we have a lot to discover in terms of these disordered ordered regions here.
Yeah.
And we also see that now with AlphaFold 3, actually some improvement also in terms of being able to move on with those regions. But
Yeah.
otherwise, I mean, you are absolutely right, Gopal, that everything I show when I say there are 72 clusters, this is of course all, let's say, defined by how we set the threshold for what we define as one cluster. So yeah.
Great. Thanks.
Alexandra, go ahead.
Hi, thanks for the talk. I really enjoyed it. I was wondering what you think about the actual pathogenicity. So, do you think that the fungi are being pathogens by affecting the microbiome, or are they targeting the plant directly? It's just that the genes are harder to identify, or the signatures of selection are more subtle.
Mhm. So I do think that there is a very important component that we still understand very poorly, which is related to interaction with the microbiome of the host. And I did not go into details with that, but we have been establishing a culture collection which is from the apoplastic fluid of the leaf. So essentially, you can sort of suck out the inside, the liquid that is in between the cells in the leaf, where you have first surface sterilized the leaf. So you are actually enriching for the microbiome that's inside the leaf. And that's also where the plant hyphae is growing. And there is a rich diversity of microorganisms. And when we put Cymoseptoria on a petri dish together with these bacteria or fungi, we can see there is a lot of antagonistic interactions. And so in the plant, the fungus must also somehow overcome such antagonists. So of course they are there; they're competing for space, for nutrients. So I do think there's an important component of that. However, evidently there is still the immune system of the plant. And so it also has to overcome the immune system of the plant. So it's I believe a combination of let's say of traits that's important for the fungus.
Let's make Richard the last question before we turn it over
One,
just to
one,
the fellows.
one quick question. Thanks, Eva. Nice to see you and great presentation. I kind of follow up on Alexandra's question. This sort of like, it's fascinating, first of all, that this whole area of these effectors having, antimicrobial activity and in your case, 50%, right? Of the,
Yes. Yeah.
I mean, that's pretty amazing, kind of a game changer. But what does it mean in terms of potential application? Is this something that we were going to be able to sort of modify the microbiomes to ensure that they're not susceptible to these antimicrobial activity, or like what are your thoughts about the future applications of these findings?
So I think there is now I haven't talked about it, but we do a lot of these. So one thing is looking at the function of proteins, but we also put are collection of microorganisms together with Septoratridae, and then we quantify these antagonistic different types of interactions. There are also beneficial interactions. And so I think there is for sure a large Septoratridae is actually inhibited by several of these bacteria. And so I think there is a potential there in terms of discovering molecules produced by these bacteria that act specifically against the fungal pathogen. So that's one side of it. But another side of it is, I think, there is, of course, the whole perspective of application of biocontrol agents, where I think working at this level with understanding these complex interactions is important for developing something that is robust and that will also survive in the field and have long-term effects against the pathogen. So I think understanding this ecological interaction is important.
Okay, so at this point, I think we'll ask all the non-fellows to leave the room, and we'll leave the fellows to have a smaller conversation. I hope you'll have questions not only about the science, but also about careers, et cetera. We've got a visitor here from Europe, so if you want to understand how research is different in Europe or whatever, you can ask all of those kinds of questions too. Do we have a fellow who's going to kind of lead this discussion?
Thank you, Eva. Hopefully see you soon.
Yeah, see you.
Bye.
Thanks for coming.
Anybody, any of the fellows willing to kind of moderate or make sure that, yeah, Christine.
Okay, Christine, thank you.
Much, yeah. Okay, then I'm going to also depart, but Eva, thanks again. That was